highly conserved chromosome centenic region. And this is the same corresponding region in the gray fox. So when we consider that in the carnivores and also human, these are highly conserved regions, we can then examine the patterns of reorganization in the gray fox and the red fox. So pointing your attention to the dog, which is part of the wolf-like grouping, these regions correspond to mature centromeres. Because we believe that this region in red fox represents a neocentromere, and those conserved regions are separated by a centromere, we believe that this establishment of a centromere exists, that this event establishing a centromere between the chromosome 13 and 29 of evolutionarily conserved chromosome segments is a newer structure. Concerning the gray fox chromosome, this association is preserved from the ancestral structures. And we believe that the reorganization that reorients these two segments may be a new event. So the neocentromere here has been established in recent evolutionary time. And the reorientation of this chromosome segment into an interstitial region is in the process of losing centromeric repeats. One of the results of this data is that we hypothesize several scenarios that might explain why the maned wolf and bush dog have different evolutionarily conserved chromosome segment patterns compared to their fellow South American canids. In the crab-eating fox and the hoary fox, they do have these two segments separated. However, these two species do conserve the association on one chromosome. And so if you track back that pattern to the last common ancestor between the wolf-like and South American canids, we have this scenario where, unlike the wolf-like and this grouping, these two species in the South American group have a contradictory formation of the chromosome segments. So what we believe may have happened is that the ancestor to these species may have undergone a change that reorganized. So the transition from an ancestral, the transition of ancestral structures would either, in two scenarios, be lost in the wolf-like and crab-eating fox / hoary fox groupings, or it may have been preserved and lost or it may have been lost and then preserved in these. So the question is whether or not the transition of grouping of these segments changed at this location and this location, or at this location and this location. And so phylogenetic trees really track these sorts of changes. What I believe happened is that the more parsimonious the more parsimonious phylogenetic tree would actually move these two species here. And so the result would mean fewer state changes. So in effect, instead of having state changes, two state changes for these two species, there would be one state change. And so this has actually not been published yet. However, I believe that this may be incorporated in further phylogenetic trees as well.